I'm Kang, and today I'm gonna talk about the efficacy-based quality control of herbal medicine. Uh, I'm talking about the efficacy because the impact, the quality control usually carried out by some analytical tools like liquid chromatography or the GC like that. As shown in this content, I'm briefly talk about the introduction of quality control, and then I'll explain the development of herbal medicinal product and its difficulty in quality control, and then I will introduce some effort carried out in China or United States, like quality markers or the connectivity maps. In final part of my talk, I'll talk about the quality control of Korean medicine as an example of So-Tang. So in the first part of my talk, I will introduce the concept of quality control. Yeah, we expect high quality of herbal medicine actually, because the high quality of herbal medicine may guarantee its efficacy. Actually, in Japan, they producing a herbal medicine with a high standard and attracting interest in China. The Japanese Kanpo medicine market, Kanpo means Korean medicine or Chinese medicine, market is over 160 billion yen, and the Jumura is selling 100 billion annually. This means that the improved or advanced quality control system may guarantee the consistency in efficacy, and also we can expect that with the high quality of whole medicine, the consumption of Korean medicine will increase, and also some companies may also increase the profits. This slide shows the quality control of pharmaceuticals, because the quality control came from the pharmaceuticals. As shown in this slide, drugs must be marketed as safe and therapeutically active formulations because it should be safe, because patient or the users may have difficulties. Also, the terms quality control refers to the sum of all procedures undertaken to ensure the identity and the purity. The pharmaceutical like aspirin I show in the bottom should be aspirin, and the purity will need to be high. According to the US pharmacopoeia method, aspirin must be contained in the range of 0.5%. And in the right side, there is a chromatogram of aspirin. So quality control usually carried out with this kind of analytical tools. This slide shows some difficulties in the quality control of herbal medicine. Herbal medicine, I will explain more detail in the later slide. The controlling of quantity of herbal medicine is very important and crucial for ensuring their efficacy and safety, but has many limitations. As you well know, they are plants. The plants are grown in the soil, so their effect and the contents may differ. Like climate, we actually had lots of rain and hot days in the summer. Maybe the ingredient may be deeper, and the soil, cultivation method, collecting time, and so on. So herbal medicine made from plant and may be deeper in the ingredient, and also their effect may be deeper changes. So we need to know that the property, the characteristic of herbal medicine is a multi-component and multi-target. This property of traditional Chinese medicine give rise to the necessity for developing a new theory. Because currently we are using a quality control of a few compound, like one or two compound can apply to the quality control. But we expect more high quality of herbal medicine. So we need a new system, new method, more scientifically good method we are expecting. However, the pharmacopoeia quality monographs of herbal medicines are few numbers of chemical-based paste, and which cannot ensure the consistency in therapeutic activity. So, I show in the slide in the left side there is aspirin. This pure one is almost 100% of aspirin. It can be quality. We can control the quality of this medicine by some analytical tool easily. In the middle of the slide, I show the Artemisia algae. We made it the herbal medicine. Strain was developed from this plant. Strain is using for the gastritis gastritis. And the right side, I show the sociotang. Yeah, sociotang. We are using intestinal disorders in our country. It made from 7 herbs, as shown here. So we expect that the quality control of styrian is difficult, but the quality control of sociotang will be more difficult. So the objective of my presentation is to share the current status of research on difficulties and solution of biological quality evaluation of herbal medicine. So actually the biological evaluation has conducted several times, but have failed because it has a lot of indication and multi-component. So to measuring the biological efficacy is also very difficult. In the second part of my talk, I will explain the development of herbal medicinal product. Natural product or raw materials can be broadly divided into cosmetics, and food supplement and herbal medicinal product. In the United States, herbal medicinal product is a botanical drug, and we use sometimes the cosmeceutical or nutraceutical because we expect some therapeutical effect. So like a pharmaceutical, we can add ceutical in here. And this slide shows the research and development scheme for herbal medicinal product. From the bottom, we need to select a candidate first. The candidate came from some literature study or feasibility study like some screening method. When we select the candidate, then the plant needs to be extracted. Extract means some kind of herbal medicine we extract with the water, and some functional food we extract with the ethanol like that. And total extract, and we divide it into sub-fraction like that. So we apply biological assay like in vitro or in vivo studies on it. So we call it activity guided fractionation. And finally, if we find the good effect of fractions or like a compound, it can be developed as a medicine. But as I shown in left side, the effect of a single active compound is often weaker than that of original herb. So original herb has a more strong effect. So it can be developed as a drug like here. So herbal medicine can be developed, but as I explained here, their contents, ingredient may vary, climate or soil or the other reasons. So optimization or standardizing activity fraction is needed. In this part, you can say that quality control is needed for this step. This paper is our work. Our work is published 1 year ago. We worked with the Stilan that I introduced in the postpartum talk. Is a herbal medicine for the gastritis. In this paper, we analyzed the active compound and also tested its effect. In the right side, we tested it with some anti-inflammatory properties like phosphorylation of MEP kinase, like nuclear factor kappa B, INOS, COX2 like that. It has an effect, but currently the quality control markers for this stirin is two compound, eupathyline and hispidolin. In this paper, we analyze gesioscidin also, but in the pharmacopoeia, it's functional food, not the pharmacopoeia. It is quality control markers are eupathyline and hispidolin. But when we tested it, this black bar is eupathyline. It has no effect. The total extract still has an effect, but the eupathyline is not effect. Just we are using eupathyline for the quality control. That has no effect, but we are using eupathyline for the quality control. And also, the content of hispidolin has some effect, but the content is under 0.07%, very low. So the there is a problem. This whole herb, very low content of ingredient are using for the quality control of stilan. So, and as shown in the HPH chromatogram is a eupathyline, some higher content of eupathyline, but has no effect, and very low content of hispidolin. And there is also, as you can see here, there is a lot of unidentified ingredient like that. Maybe this ingredient may explain the efficacy of stilan. So current QC, quality control system, has some problem, I think. And how about the Soshotang? Soshotang contains 7 herbs, as shown here. The Korean Herbal Pharmacopoeia, KHP, mandates that in 1 gram of dry matter of Soshotang should be contained 1.6 milligram of glyceric acid and 11.9 milligram of bicaline. Currently, just two compounds, glyceric acid and bicaline, are using for the quality control of Soshotang. Do you think? I think it's a problem. One herb, the Artemisia algae, stirin, using two compound of quality control marker. Soshotang, we expect more compound, maybe multi-component and multi-target, but we are using just 2 markers for the quality control. It has some problem. So also in Yale University, Dr. Chang also insisting that the current chemical markers are irrelevant. That means not suitable. He's emphasizing that the importance of use of suitable biological assay on its mechanism of action should be developed for quality control. So this is the summary for the second part of my talk. Currently, we have some limitation of quality control system of herbal medicine because although the herbal medicine have a lot of herb, but the single or few index components are applied for the quality control of herbal medicine. And the other thing is, in many cases, the active ingredient cannot be found even after activity-guided fractionation. That is, the correlating the efficacy and the chemical characteristics is difficult now. So the other countries, China and United States, also has also thinking about that the current quality control system has some problem. So they are making effort, people us. So I'm gonna introduce some example of research technique carried out in the China and the United States. Quality markers in the China, they make some good name of Q marker. In the traditional Chinese medicine, the research is underway to utilize some chemical component contained herbal medicine, and the Q marker, they named it. The characteristic of Q marker is a characteristic is a 3-point. First one is quantitatively verifiable. It can be, we need to analyze it. And second one is it should be related to activity. And third one is a specific component in it. So they should be contained in it. So these 3 points are very important for the Q markers. This slide looks very confusing, but I will explain briefly. In the most Q marker studies, it should be started with a strict preparation of current chemical-based QC markers. So it should be identified and sometimes use DNA barcoding for the identification of plant and also processing the slice or cutting, drying like that. It should be strictly followed it, and common quantitative determination of LC or GC are conducted. And this is a new point of Q marker. In this theory, they're using a property of herbal medicine, multi-component or taste of herbal medicine, and the effect is also important. And third one is component. These 3 kind of concept should be followed in these Q markers, and the multi-discipline based strategy. That means for the one goal, all of the research specialists are working on together, like chemists, like natural product chemistry, analytical chemistry, bionics are analyzing the active compound, and the computer-aided design and system biology expect some active mechanism and genes, and actually the system biology is a very powerful tool to explain the multi-component or multi-target of characteristics of herbal medicine. And the pharmacologists like me, and finally confirm the activity of this compound. This is the example of some Q marker study using a Corydalis rhizoma. As usual, the chromatogram of Corydalis rhizoma, we can identify the 28 alkaloid like that, many alkaloid, and they define the potential Q markers of Corydalis rhizoma in this plant. This kind of 28 alkaloid can be synthesized in this plant only, and they define that specificity. That means this kind of alkaloid are specifically contained in this plant. And then also they experimented with the antalgic effect of this plant with 60% ethanol extract, and using a network pharmacology. Recently, we are using AI or lots of OpenDB based studies. So I think you can understand some network analysis. So in brief, we use a computer to expect some pathway or mechanism, the multi-component using this tool. So this is a powerful tool for the analysis of multi-component. And then multidiscipline is very important. So chemist and and some kind of dry lab, or we can also docking it, and there is some property. In the Korean medicine, we importantly thinking about that the taste, bitter or pungent taste, can be related to some receptor. So we can expect some receptors involved in using a molecular docking. And the important thing is, as I indicated in the red character, they detected in the blood this compound, and also they also detected in the brain, can be detected. So they proved that these compound alkaloid are observed in the body and go to the brain and antalgic effect. So these 7 alkaloid were identified as Q markers, and we can explain more accurately quality control. And the second thing is about the connectivity map. Connectivity is connect means a relationship with each other. In the United States, there was some research project carried out in the Broad Institute in the LINKS program. It was supported by NIH, and in this program, they collect lots of omics data, like genomic or largely gene-related data. I will explain more detail in next slide. So in the powerful tool was L1000 array. L1000 array is L1000 means in brief, with just 10 transcriptome, just 10, we can expect to 12,000 expression. So L1000 means just with a small amount of gene may effect on the other gene. Usually, there are several kinds of pathways and lots of genes. In the microarray, there are lots of hundreds of thousands of lots of genes. But in the signal pathway, there may be upper genes can control the lower gene. So just 10 transcriptome gene expression, we can expect more than 10,000 genes. 10 means L1000 array. And this method is explained by the landmark transcription. Landmark means like a landmark. We can understand the landmark. In this city, there is a landmark building, landmark spot like that. So some landmark of gene can control the lower level genes. And this slide shows the principle of connectivity map. Connectivity map means using a correlation structure. I'll explain briefly in here. There is a compound with unknown mechanism of action, like genistein. We actually know genistein, but we assume that this compound has an unknown mechanism. We can treat it in the cell and extract it and analysis with the gene expression, like here. And there will be a change in the gene expression. Some genes may be upregulated, and some genes may be not changed, and the other gene may be downregulated. With this characteristic, we can put it in the database, and we can get lots of information. There may be a similar in the gene expression in the compound, and we can expect that if the genistein in here, and some related compound may be located near here. So we can expect that genistein has a similar effect in the other cell, like that. This is a concept of correlation structure and connectivity maps. And the final part of my talk, I will explain the quality control of Korean medicine with the main topic of this presentation. We are currently working on the several kinds of herbal medicine, and the social tang is one of example. Social tang is Chinese medicine, a Chinese name, and has been used for the fever, stomatitis, and gastrointestinal disorders in Asia. And its classified code in Korean FDA is other digestive tract drugs. And its authorized efficacy is nausea, loss of appetite, and gastritis. And the other gastrointestinal weakness, fatigue, and late symptom of cold. In the upper part of efficacies can be measured easily, but in the lower side of symptoms cannot be tested easy because we cannot accurately test the late symptom of cold. There is a drug for cold, but what is the late symptom of cold? We cannot understand easily. So, as I explained before, Socio-Tang is currently quality controlled by just two compounds. So, quality control through single few index compound is a problem. It cannot explain the multi-mechanism or polypharmacology effect of Socio-Tang. And also, Socio-Tang has lots of efficacies. We recently published a review paper. I will explain later. In the paper, we could identify that the use of Socio-Tang in 3 countries, China, Japan, and Korea, is slightly deeper. In Japan, Socio-Tang are used for some kind of cirrhosis or hepatotoxicity, protection of hepatotoxicity. In the China, it's using for some depression. In Korea, we are using gastrointestinal disorders. So the defining this prescription, the effect may be deeper very much. So we using the purpose of Korean medicine is not cure the specific target or drug, the target or disease. It's a syndrome-like disease. So set of symptoms make it difficult to pathological mechanism and hard to understand or explain by the simple method. This paper has published last month. We studied it. In this paper, we try to explain the multi-target herbal medicine prescription and its therapeutic biomarkers in it. So we analyzed several cell type and treatment dosage and significant markers in here. There is lots of inflammatory signal pathways. And in this small figure, as I explained before, this paper is clinical studies. In here, first one is our the RCT conducted in Korea. It's using a gastrointestinal disorder, and second one, depression, was carried out in the China, and fourth one, they use a chronic hepatitis in Japan. Socio-Tang are using for the treatment of chronic hepatitis. So in 3 countries, the use of Socio-Tang is deeper. And we summarized that the Socio-Tang's markers, as shown here, it can be applied to cancer. It is using for cancer, liver fibrosis, and infection like other metabolic disease. So for this literature study, we have a lessons from this study. The suitable indication, because in 3 countries, we are using a different usage. So suitable indication of herbal medicine can be identified from the clinical and in vivo studies. We are using lots of money for the clinical studies, so it should be we need to think more about it. And so clinical study, we can identify some suitable indication. And the quality evaluation biomarkers can be a set of some in vitro experiment, and should be explain the mechanism of actions. And these biomarkers must reflect the multi-component and multi-target property of Korean medicine. And we are now working on to select the optimal relevant biomarkers from it. So current quality control of herbal medicine, there is a problem because, for example, 소시오탕 has several kind of several effect, like fever or pain can be tested easily, like inflammation or using a raw cell, we can measure the inhibition of nitric oxide like that. But on the right side, the gastrointestinal weakness or lay symptoms cold cannot be measured easily. So we need to redefine the disease, redefine the means because if we don't redefine in it, the measure method, maybe there are lots of methods. We cannot choose it easily. So redefining is needed, and we need to investigate the genes associated with each symptom of indication. So we can apply the L1000 study in here like that. There are lots of components in the social tank. We can select it from the open database. Like there are lots of analysis results, and we can choose the bioavailable compound first. It should be observed in the body. After selecting them, and we can using network pharmacological experiment, we can expect some genes related to social tank. And we can find out the landmark gene. I mean, upper level of genes can control the other one. By controlling upper level of gene and measuring it, we can expect it's a relevant one, suitable biomarkers. This is the final part of my talk. So currently, there are several limitations. Current chemical marker-based QC is insufficient, as I explained before. Just a few compounds cannot explain the multi-component and multi-target property of Korean medicine. And chemical marker-based QC is difficult, but the efficacy evaluation-based QC is even more difficult. So the strategy for the success is as conducted in China using a Q marker. We also should work on establishment of combinatorial strategy. That means a multidisciplinary study. For the one goal, we should collaborate together. Like biologists should work with chemists, and also need to use recent big data analysis and AI technology. Actually, there is lots of OpenDB is free. Using a string CMap like that, we can expect and we can get much result in it. So multidiscipline-based strategy is needed. And the second one is because the herbal medicine, herbal formula is contained with lots of herb. So we should take in mind that the quality control should be conducted with a polypharmacological approach, using a multiple target and multiple indicated component. Can overcome the limitation of current QC system. Okay, these are lab members, our lab members, and thank you for your attention.